Okay, so when it comes to diagnosing herpes viruses, basically this can be, of course, usually it's quite easy and simple. You look at basically just these vesicular lesions. As I said, they're very characteristic in this kind of sense and how they progress. But of course, there's many different laboratory confirmations you can do. For example, you can take, if you have a Saznak smear, this is a smear where you take basically cells from where you see the vesicles forming and you look at them. You look at under a microscope. Do you see basically these multinucleated giant cells? Do we get these? Because remember, herpes viruses, what they do, they love to basically do these multinucleated cells. They really love to do that, kind of bring them all together. So if you do see multinucleated cells, that tells you that it's herpes viruses. But of course, this does not allow to know specifically what type of HSV, but also are not able to distinguish between HSV and varicella zoster virus. Because one thing that we're going to see with varicella zoster virus is that it has a very similar, let's say, kind of looking vesicles that they form. So sometimes, if you want to distinguish them, this is not the easiest thing because the varicella zoster virus also can induce basically these multinucleated cells. So the best thing is using PCR. So PCR, it really helps you distinguish between HSV and varicella zoster virus, but of course, it helps you to understand which one do you have. Do you have HSV1 or do you have HSV2? And in specifically, it's so standardized. It's the golden method. Basically, majority of all it or not if all basically hospitals have already a standard procedure in how basically to do this. PCR is amazing. It allows also for detection of viral DNA even when the viral load is very low or when there's not even lesions visible because in per se to get the pap smear, if you want to see this, you need to have a very, let's say, high infection, high viral titers to either actually see this. In the case of PCR, it's super sensitive. So you can basically, the virus could be at very low levels and you can still detect it because PCR needs literally nanograms of DNA. It's very little that you need per se. So overview of antiviral treatments. Technically, you're trying to primarily stop the viral DNA from replicating. And in this sense, you can never eliminate the virus. You're not able to genetically remove the virus DNA from your own genome. So that doesn't happen. So you can only basically stop its replication. How do you do that? Is by basically using nucleoside analogs. And one of them that I spoke in my last lecture was acyclovir, which I'm not going to go into details because I've already explained this. So now let's go into another very interesting type of herpes viruses, which is still within the alpha family, which is the varicella zoster virus. So very similar to the HSV, structurally very similar and biological, has many different similarities. Them, of course, being enveloped, its linear double-strand DNA genome. It replicates in also in the nucleus. But you think about other herpes viruses, the VSV establishes latency. So after you get infected the first time, you get your symptoms, and then you get the latency. And then the latency basically here can take usually takes can take much more longer than basically the herpes simplex viruses, meaning that to get reactivated, it usually takes years, quite a lot compared to the herpes simplex viruses. So the first time you get infected with basically the VSV, you get varicella, chickenpox. We know this. This is basically a very contagious and typically occurring disease during our childhood, where you get basically what happens in some way, the virus literally gets through into our blood, which then is able to literally go into our skin, and they create this really kind of vesicular rash where you have these little vesicles, and you get this successive crops, which basically means that these are lesions at different development stages that are happening at the same time. So if you look at this baby over here, you have these really big kind of vesicles already forming, they're already about to pop, while you have these other very small ones still growing. So this is what we mean by successive crops. Simultaneously, you have many different types of vesicles, all at different stages of growing. So this is very, let's say, indicative of varicella or chickenpox. So usually, basically, the infection, as I said, it happens in the respiratory tract. It goes into our bloodstream, then to our skin. Usually, it's extremely mild in children. We've all gotten varicella or chickenpox, or the majority of us have gotten it. But where it can be very dangerous is in adults and in immunocompromised individuals. Varicella, when we get affected as children, for some reason, we do not get much of a symptom. Like we do get these symptoms, but that's it. When it comes to adults, the symptoms of varicella or chickenpox are extreme. They are much more neurological and can be very dangerous. And of course, if you're also immunocompromised, the virus can really disseminate into many organs that can cause death. So basically, similar to your herpes simplex virus, the zoster virus, the VSV, it will basically establish latency. It's going to go to, again, a sensory nerve ganglia, and there it's going to do the exact same thing and literally insert its genome, and then it's going to stop replication. And it can basically be years or decades, literally 20, 30 years later on, from the first time that you got it, let's imagine at 5 years old, that later at 50, so 45 years later, that this virus suddenly wakes up. And it wakes up and basically it leads to herpes zoster. Also, we call this shingles. So, where we really basically again get this rashes all over basically, you know, our skin. And you basically have these, let's say, localized vesicular rashes. So their distribution is at a certain space, and this basically it what kind of reflects is the involvement of specific nerves. So if the nerve over here is basically, let's say, infected, you're gonna get basically in this certain area this kind of rash. So where it basically forms its latency, you start to see where the vesicular rash is localized. And again, you get very similar to what you see with herpes simplex viruses. You have this erythematosus base, so this bed over here. Then you have these vesicles. Again, very similar, these kind of sacs filled with water that will pop and will literally crust over throughout the time. So this is why sometimes, in general, seeing them, sometimes you can get confused between varicella zoster and the herpes simplex viruses per se. The issue with this is that this is nothing. Sure, you get this and you get reactivated. This is manageable. The issue with, and generally with herpes zoster or shingles, is basically this post-therapeutic neuralgia. This post-therapeutic neuralgia is basically a condition in where pain persists even when the rash has been actually resolved and, let's say, kind of cleared off. This is a pain, we call this a ghost pain that stays there. And you basically are in constant pain for quite a long time. This can be extremely debilitating, and this can really kind of cause a lot of people to stop working and enter depression because you're constantly in pain. So it's very kind of annoying and a very life-threatening disease. More not life-threatening, more like debilitating disease that can really put you at home and basically be unable to do many different functions because you're constantly in pain. So when you look at the complications, and generally with chickenpox, usually it's very mild, it's self-limiting in children, you don't get much. But the complication happens much more in adults, specifically also in pregnant women and immunocompromised individuals. Because what you get, of course, with varicella or chickenpox is you basically, of course, you these vesicles, they're literally doing a lot of openings in your skin. This allows secondary bacterial infections to get inside and cause many different types of infections, specifically cellulitis and systemic infections. But of course, the varicella, the varicella zoster viruses, they can go inside to your lungs, causing pneumonia, but they can also go into our literally brain to cause a lot of neurological complications. So like encephalitis or cerebral ataxia. Cerebral ataxia is basically death of certain areas in your cerebellum. Of course, the complication of herpes zoster. So basically, when it basically this is coming after, this is the recurrence after the late it goes into latency and it gets reactivated. You get herpes zoster or shingles. This basically the complications of this is simply, of course, we know is you start to actually have, as we know, this post-therapeutic neuralgia, which technically causes a lot of pain. But of course, other than that, we can also get a lot of issues. The virus can still go into other nerves, specifically your ocular nerves, that can cause a lot of issues with our eyesight to the point that can leave us almost blind. So completely leading to vision loss. So the immune response to varicella zoster virus, as we know, basically, we do have both humoral and cellular immunities. We also do have your innate immune system coming to attack. but it's usually the T cells. It's usually T cells that are super important. And what we technically see, specifically with people that are suddenly get, because all of us have had, well, not all of us, but let's say a overwhelming majority of us have had, chickenpox. But why, as only a handful of people get the shingles or herpes zoster, is mostly these people have some issues with their T cells. So T cells are extremely important in keeping this virus in check. If you have some sort of basically you're immunocompromised, you have AIDS, you are under a transplant, or basically you have a transplant of an organ, and you're under basically compromising drugs, then this virus tends to wake up. So the best thing to do in generally is that when you hit a certain age, specifically at 50, because around 50 is the time that your immune system starts to fall down, your basically this varicella virus starts to wake up and cause shingles. So in these ones, we like to give basically these chickenpox vaccines. So the varicella zoster vaccine, very important. It really helps and boosters your T cells and your B cells to keep this virus in check and not to get reactivated. Now going into other types of viruses, the Epstein-Barr virus, it's one of the most common viruses worldwide. It's actually transmitted through saliva, and because it's transmitted through saliva, we call it the kissing disease because the best mode of transmission is through kissing. So I'm quite sure you've heard this in schools, basically the kissing disease. So usually Epstein-Barr viruses, it can actually infect epithelial, but it loves to infect B cells. And there it can start and establishes a lifelong latent infection. So once you're infected with EBV, it stays there for your life. So like other herpes viruses, the EPV basically goes from latent to lytic phases. So they basically can go, you know, and generally start causing disease or symptoms and then basically be just dormant. So what it does, of course, is it usually enters through your oropharynx kind of epithelial cells. But once it basically infects your epithelial cells, they start to produce so many viruses that they get out and they basically bind to CD21, which is found on B cells. Once in the B cells, it loves to stay there. It does the latency over there, and basically it will stay there for your lifetime. And there, what it can do is it can produce and promote cell survival and proliferation. So it can really mess up the B cells kind of proliferation signaling and stop its apoptosis. And this can cause actually Burkitt's lymphoma, which is basically, as we said, is a type of basic cancer in your B cells. So the first time you get actually infected with EBV, we develop something called mononucleosis. So very typical in teenagers when basically we start kissing each other, but also in young adults. So what we see basically is the first time you get infected with EBV, you develop mononucleosis. It's a disease that's characterized by fever, by sore throat, lymphadenopathy, so where your lymph nodes are being basically quite swollen, and fatigueness. What we see that is very a big hallmark of mononucleosis, because mononucleosis is specifically ABV, when it infects B cells, what it does, it really gets T cells quite activated. So we see this atypical lymphocytes. By that I mean, these are your T cells. They look very nice and round, but during let's say, mononucleosis kind of disease, or when your ABV is basically active, it causes these T cells to have this atypical shape. So rather than this nice singular kind of cells, they're very kind of, let's say, in some sort of way, very morphologically distinct and bigger. And this is actually quite a good thing. This is actually what helps the EBV to stay in check. They're much more activated. But because these T cells are so active, these are the ones that are causing us to be fatigued, to actually have fever, but also have these kind of swollen lymph nodes. It's actually the atypical lymphocytes that causes these symptoms within us. But of course, once it starts the latency, what basically happens there, the EBV is going to keep literally your B cells to keep proliferating, sometimes causing Burkitt's lymphoma. But can also basically, of course, produce latency in epithelial cells, so it can actually cause also certain, cancers in the nasopharyngeal in the nasopharyngeal kind of tissues, so causing there the carcinoma. So you see a lot of EBV basically based cancers when you're immunocompromised. So the virus starts to really go unchecked over there. So as we know, the EBV has this very nice latency. So once it's basically in your B cells, what it does, it's able to express a lot of these different small different types of it expresses limited number of genes, but majority of these genes that are being expressed, they're mostly there to evade immune detection. There, it's helping basically immune cells not to really evade it. To it helps the immune system not to be able to kind of recognize it. So because it's not able to be recognized, it's then much more easier to keep being transmitted. And of course, this is basically transmitted through the shedding in its saliva. So now let's talk about cytomegaloviruses, CMV. CMV again is one of the most common viruses as well. Very similar to your Epstein-Barr viruses, meaning that they basically also have this lifelong latent infections. They are basically enveloped, linear, double-stranded DNA genomes. So CMV is extremely common, and usually CMV, the first time we get basically infected, we do develop symptoms, but very, very basically rarely. But usually when we get the CMV, it goes into this latency, and usually in latency, it doesn't do much unless you're immunocompromised. If you're not immunocompromised, usually CMV does not do much. It's it's not as much as big of a killer like your EBV. So in my kind of let's say experience, it's quite hard to see people dying from CMV unless you are immunocompromised. Then that can be a life-threatening disease. So as I said, usually CMV infection is usually mild or very asymptomatic. If you do have symptoms, they're very resembling of mononucleosis. You have fever, fatigue, and lymphadenopathy. But one thing that people use to, let's say, kind of distinguish between mono and CMV infections, Epstein virus infection, so in mono, you do get sore throat. CMV, you do not. So that's usually how you're able to kind of, let's say, distinguish. Thing about CMV, it basically it's able to do basically its latency, and it's able to do it in almost every type of cell. It's quite weird in that kind of sense. So what it basically does in there, the CMV, when it's in its latency, usually if you have a good immune system, it doesn't do nothing. It's only when you are immunocompromised that it starts to affect so many different types of basically organs. It can affect your brain, it can affect your lungs, your liver, your kidneys, your stomach, you name it. It could be quite a lot of stuff that actually can happen. But of course, one thing that CMV does is there's congenital infections. So usually mothers can pass on CMV to their kids, and this can be extremely dangerous because if CMV does manage to actually be transmitted to kids, it can cause a lot of the congenital defects. Specifically, this we call this microcephaly, where basically the let's say the head in some sort of way of the baby is not properly developed. It's under its typical head size, and this can cause a lot of different types of issues later on during the child's development. But it can also cause a lot of different other consequences. For example, it can cause actually hearing loss. So usually a lot of the hearing loss when it comes to kids is usually can be done through a CMV viruses. Because you have to remember that babies, if they're infected CMV, usually their immune system is still not that good to stop it. So now let's go on to human papillomaviruses. The human papillomaviruses, they're a bit in some sort of way weird that these basically are non-enveloped DNA viruses. They have a circular double-stranded genome, so we moved away now from your herpes viruses. It's the most, it is the most common sexually transmitted disease worldwide. So HPV basically can be transmitted sexually, basically, but it can cause different types of lesions. The lesions can be going from benign warts, so these very weird looking kind of basically flesh like kind of lesions that I'll show you some pictures about, but they can also cause tumors. So what makes them basically cause either a wart or a tumor depends on the type of HPV. There's many different types. They're basically similar to your herpes simplex 1 and 2. These types are their serotypes. They differ basically in certain antigens in their proteins and basically in the surface. So you have certain basically papillomaviruses that only produce warts. So what happens there once they basically you are infected with HPV. HPV, what's going to do is literally going to go into your keratinocytes, and it's going to cause them to start really proliferating like crazy, but benignly, meaning that it's going to proliferate into a certain time, it's going to stop. So hence, that's why we say benign. It's not oncogenic, that meaning it's not going to keep proliferating basically forever. It's going to stop. But what you do get is there's basically this massive cell divisions and thickening of these hyperkeratotic lesions. So you have a lot of these keratinocytes that have been literally in some sort of way divided so many times, and you get the formation of these kind of skin lesions. So these we call these as warts. These are generally very harmless. They can be persistent and may require treatment. Sometimes they can go on their own. Sometimes they can stay and keep multiplying. And it's quite simple in a sense. You just basically put some medication, or by basically you can freeze them and kill them off. The issue with HPV is that it causes cancer. And usually the types of ones HPV that can cause cancer, they have basically examples are HPV 16 and 18. These guys, they have certain proteins, specifically E6 and E7. And what E6 and E7 do is they literally inactivate retinoblastoma, or so that's what E7 does. E6 promotes the degradation of P53. P53 and retinoblastoma, we call these the guardian angels of our cells. These are extremely important in inducing apoptosis. If the cell is transformed to become cancerous, P53 and retinoblastoma, what it does, they start inducing apoptosis. But in the case of HPV, their proteins E6 and E7, they stop it. So now the cell has this weird apoptotic signaling, so it doesn't have apoptosis. So they keep literally growing and growing and growing. So HPV usually causes a lot of cancers in the cervical area. So cervical cancer, it's 99% of cervical cancers come from HPV, but they can also cause cancers in the anus, in the oropharynx, and also other genital sites. So because of this, we are basically created a vaccination against HPV. So this HPV really helps us prevent 90% of all strains that can cause cervical cancers, genital warts, mouth and throat cancers, and anal cancer. So this has a huge, tremendous, actually, effect on reducing or even eliminating, in the case of cervical cancer, basically the existence of these cancers. So these are extremely important. So these HPV vaccines, they protect against many different types of HPV, be it high risk or low risk. So low risk causing the warts, and high risk causing basically the cancer. And the final viruses that I'm going to be talking about today are the pox viruses. These are very weird viruses, very unique. They have a really weird helical kind of shape. They're one of the most biggest viruses, and they're able to literally replicate entirely in the cytoplasm. And one of the viruses that I'm going to be talking today about is this Molluscum contagiosum virus, because it's the most common viruses that we see within humans. So Molluscum contagiosum is usually very harmless. We usually get this basically through, in fact, basically we can get it through skin contact. This could be technically from, touching a skin that has molluscum contagiosum, a towel, or humid kind of surfaces. So that's where we basically get them. And when we get basically these kind of molluscum contagiosum, what they do, is create these lesions. These are very different to what you see with varicella zoster or with herpes simplex or warts. These guys are very small. They're very firm, flesh-colored papules. So they have a very distinct central umbilication, meaning that the center has this indentation, so small little kind of hole. So hence it looks like you've removed the umbilical cord there. This is what we mean by umbilication. And usually they become inflamed or itchy. They usually can occur in the face or the trunk or the extremity of children, but they can also come in the genital regions. So they can basically also be transmitted sexually. So it's skin to skin contact, or of course through it can be basically just normal touch, but also could be through sexual transmission. And usually in the pathogens of molluscum, you get a lot of these inclusion bodies. So what you can do is you can remove a bit of these basically kind of, let's say, papules, and you look at basically the cells. If you see these Henderson Patterson bodies, these are these kind of large inclusion bodies inside a cell, you can already say that this is Molluscum contagiosum. So these bodies are very, let's say, characteristic of Molluscum contagiosum. So to transmission, usually it's transmitted, as I said, through skin to skin contact, but also through contact through contaminated objects like towels or clothing. This could also be through basically as well through sexual contact. Usually, the clinical course, molluscum contagiosum, is usually self-limiting disease. It usually resolves on its own spontaneously over months or a few years. So usually you don't do anything about them. They're usually benign and do not need any treatments. However, usually what happens, usually when you get molluscum contagiosum and you get this quite often, this is indicative that you have actually an issue with your immune system. A lot of immunocompromised, specifically people that have primary immunodeficiencies, what we tend to see with these people is they actually have a lot of molluscum contagiosum. So usually when we see people getting a lot of molluscum contagiosum, we tend to see that these guys have some sort of defect, specifically in their T cells. So for us, we really don't really care about molluscum because again, it's self-limiting, it's harmless. You can remove it if you want to for let's say this is more physical reasons, but in generally it doesn't really much need much more.